So now that we've gotten into DNA and we kind of know what it is and what it's used for, let's talk about how we make more of it, specifically getting into chromosomes and cell division. So why do we care? This is a really organized, highly controlled process called mitosis. And this is what's going to allow us, as well as other multicellular organisms, to grow and repair. So the growth that you experience from birth until now and the repair that you go through when you get injured are both due to the process of mitosis. And so we see that here, right? Here's one of those plants. It's growing a root system as well as going upward, but we're going to look at the root. And we see at the tip of the root is where we see a lot of cell division happening because these roots are actually growing outward and they need to make a lot of new cells in order to do that. Or let's say if you're shaving, right, shaving your face or shaving your legs, when you do that, you could be damaging cells that have to be replaced. So how do cells know when to die? Most cells are programmed with a specific endpoint in mind. If that does not happen, if the cell keeps going, that's when we have things like cancer. But we'll get into that a little bit more when we talk about the cell cycle. Let's get into DNA itself though and how we organize based on who we are. Depending on if you're a prokaryote or a eukaryote, you're going to organize your DNA differently. Let's look at the prokaryotic cells first and how they organize their DNA into a single circular chromosome. When we have to go through division, we're going to replicate in a process known as binary fission. Binary fission is where we are going to copy all of the DNA into two identical copies, and we're also going to divide the cell into two. During this process, the cell is growing, and then that parent cell, also known as the original cell, is going to pinch into two new cells that we call daughter cells. So here's that prokaryotic cell going through the process of binary fission, one bacteria and another bacterium, and inside of these guys they have one single circular chromosome that's not visible here that went through the process of replication, and then we're forming this kind of little plate as we divide into two through the process of binary fission. So let's actually look at that a little bit easier and a little bit more clear fashion with this nice drawing. So here's our parent cell, some sort of bacterium. And we have the DNA in a circular chromosome. This replication process of replicating the DNA is where we're going to basically unzip at one point, at a starting point or an origin. And as we unzip, we're going to use an enzyme to add on the nucleic acids or add on the nucleotides on each side. So as we split the two strands away from each other from the DNA, we're going to add on the new nucleotides so that we have two complete circles at the end. Once we've done that, the cell is going to elongate. We start to see this pinching in. We see that one circular chromosome's on one side and the other circular chromosome's on the opposite. And then finally we can split into two, creating the two daughter cells that are 100% identical to each other, genetically speaking. So binary fission is a form of asexual reproduction because we don't actually need any sort of partner to do this, and the cell is basically just a clone, right? This is like a way to clone yourself, so to speak. And this happens very fast. With prokaryotic cells like bacteria, we can divide in as little as 20 minutes. So you think about how fast that is. In the time it takes for you to watch these videos, those prokaryotic cells have probably gone through one or two divisions. Now eukaryotic cells have multiple chromosomes, and all of those chromosomes are in a linear format, not a circular one. And they're made of what we call chromatin. Chromatin is the DNA plus some associated proteins, and that's what's going to help these chromosomes become very compact. Because the genomes can be really, really large in eukaryotic cells, it's important that we can compact that DNA down into a much more manageable size. So let's compare the prokaryotic and eukaryotic cells in their chromosomes. We see that in the prokaryotic cells, we have one single chromosome and it is circular. And if we look at the eukaryotic side, they're linear chromosomes, and there's many of them, within a nucleus. Right? And so these histones here are those proteins that the DNA kind of winds and wraps around, compacting it down tighter and tighter.